So my lab does do as a part of the resource, a kind of distinctly different experiment that we call targeted quantitative proteomics. And this just are the folks in my lab here. Um, and So these are the folks in the lab, and I am at the Oklahoma Medical Research Foundation in Oklahoma City, Oklahoma, and my affiliation is in the Aging and Metabolism program. and so we've been hearing a lot about this idea of discovery proteomics, and you know the general idea there is you have a system, you have a biological observation, and you want to start to explore what might contribute to that biological observation. And I recall somebody yesterday talking about, you know, newborn infants at 7 days and 28 days and things like that. You know, what I always say is, as y'all come and talk to us, we understand that you see a biological question there. You have an observation that you want to explore. We may not quite totally always understand it, but we understand, you know, that you see something, you want to explore that, and proteomics is one tool that you can use. And a lot of times the thought is here about this discovery proteomics. And, you know, but within that, you may also have a hypothesis about your system and what's different in that. And you want to probe expression of a specific set of target proteins. And my example there is changes in a known biochemical pathway or protein complex. You know, essentially, your hypothesis about why you're seeing the biological differences that you're seeing. Western blot's the traditional tool for this, and it's an excellent experiment in the right hands with the right antibodies, but it can also have some weaknesses. And you know, so usually I stop right here and kind of ask, you know, how many of y'all, and usually we'd all be in one room, how many of y'all use Western blot in your work? And then kind of the follow-up question to that is, you know, where do you get your antibodies? And in particular, has anybody had a custom antibody made? And so kind of what is the point to that question is that Western blot is a targeted immunochemical method. And so what we're talking about, and a lot of the thinking that goes into designing that experiment goes into our targeted mass spectrometry experiment. But with these advantages that the development's much faster, as in an hour or two, we can multiplex, put lots of measurements into the same experiment. The results that come out are actually a number. Validation is an inherent part of the method design. And then most importantly, once developed, they're good forever and can be transferred over to other labs. And so the thinking is the same, but some of the results are slightly different. My lab uses two different methods. Kind of the banner method for these selected experiments called selected reaction monitoring. We do that on a triple quadrupole instrument in our lab. The acronym for that is SRM. And then we also do lots of this high resolution accurate mass selected ion monitoring, or SIM, that we do on an Orbitrap instrument like we heard about yesterday. Both digest the proteins down to peptides, and then within those digests, we select peptides from each protein to go in and use the mass spectrometer to go in and measure those specific peptides as quantitative markers of the parent protein. They're fundamentally LC experiments. So we're looking at chromatographic retention time and peak shape to tell us, you know, what it is. And then the area under the curve tells us how much of it's there. And the role of the mass spectrometer, in this case, you know, they always bring sensitivity to the game, but also this specificity of analysis to know that we're detecting what we think we're detecting. So we'll just kind of look at this. This is a high resolution accurate mass experiment. It's about the simplest LCMS experiment we can do. We run the sample out over time with a gradient elution. We just scan from 300 to 1100 M over Z throughout it. We record about 3,000 spectra. And, as we look at these data, we can go in and pull out any spectrum at any time. And this is just one of them. And the point to here is, Sam just sort of invoked this idea of dynamic range, is we have some high abundance things, but as we look down into the graphs here, we can see even things that are 10% and lower, we can measure with good mass accuracy. We can see the isotope cluster. We can tell that this is a doubly charged ion. And so these signals are in there, going from things that are, maybe 1% of the most abundant, all the way up to the most abundant things in there. And this is kind of another way to look at it. So this is a mouse something, I forget what tissue this is, but you see time on the x-axis, mass to charge on the y-axis, and essentially every spot here represents a signal. And I'll point out here, we don't try and interpret this ever. This is just to kind of show you, or give you a sense of how complex these things are. And just as a reminder, the mouse proteome is known. There are about 30,000 proteins, 60 million amino acids, about 2 million peptides in a perfect tryptic digest. And so in a sense, we already know the answer because we know the proteome. We just don't quite know where that answer lies up here. The way we get at that is we kind of ask these sort of Alexa type questions of, within this set of data, Alexa, show me where 647.8537 is, because I know that's the molecular ion for this particular peptide, and then we detect it there. Area under the curve is 24,000 million, so this is a big signal of an abundant protein. Same thing for the other peptide. Show me where 849.4565 is, and it's pulled out of this complex data set these two specific peptides that are markers for acyl-CoA oxidase, and this is a liver sample, and so we call this selected ion monitoring, and these peptides become our assay for acyl-CoA oxidase. The same fragmentation pattern information that tells us about the identity, we can leverage that for this selected reaction monitoring. So the reaction in SRM are fragmentation reactions. And so the molecular ion is relatively characteristic or is characteristic of this particular peptide. We saw that in the other analysis. But then this fragmentation pattern is also characteristic of this protein. And so we can leverage that for our selected reaction monitoring experiment. And so to detect the SFL peptide, we program the triple quadrupole instrument to select this ion in the first half of the instrument, of the tandem instrument, fragment it, and then collect these fragment ions that you see in this spectrum here in the second half of the tandem instrument. And again, we get a nice clean chromatographic peak where the retention time, and this information would be characteristic of the peptide, and the area under the curve tells us how much of it's there. So same thing for the other peptide. And this is what the data look like. We look at our data using a program called
Skyline.
Skyline, thank you. And so, and what you see here is the predicted time. So once we learn this, we enter these things into a database, and then Skyline helps us to interpret our data. And so it breaks it down with the different abundances, and these abundances and this color pattern would be characteristic of this peptide, and also helps us find the correct chromatographic peak. And specificity, 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 this is what we're after. We're after making sure that this signal is due to this peptide, which we know is due to acyl-CoA oxidase 1. So where do these assays come from? I always emphasize that they're carefully designed, and they're based on things we already know about the protein. We know the amino acid sequence. We have databases out there, public domain databases, and one's called the Peptide Atlas, where we can go and see what the mass spectrometry community has detected for these different proteins. We can calculate the retention times with an internet calculator that's out there. And something that I use as a part of the development process to sort of see what to expect is this database of mRNA expression data that I can get through the BioGPS, and that's what you see here. And just kind of the point here is that my lab, my arrows are off just a tiny bit, but my lab's interested in heart, and so it's down here at maybe 3,000, 2,000 or 3,000 expression numbers, whatever those are. Whereas in liver, it's up over 16,000, and so it's much more abundant here. I can develop my assay in liver and then apply it over here in heart. And so we know we've developed it correctly, and now we're beginning to apply it. And that's something we come back to always, is we maintain a tissue library, a sample library to help us with these method development experiments. And just a point here, it's not a hard process. I teach people to do this all the time. And it's reasonable to expect that if we start the day with a list of 5 or 6 or 7 proteins and a good sample, by the end of the day, it's pretty straightforward to have 5 or so working assays. So this is what we take out of Peptide Atlas. These are the peptides that people have seen. We use this as the starting point for our design process. I have a few trouble amino acids that I tend to avoid. We also tend to avoid peptides that are really hydrophobic or too large for various reasons. And the key thing we need along the way is a good sample where the peptides are detectable, because most of our development is done with endogenous amounts of protein. So we take this list of peptides and we test them experimentally. And so here they are, calculated M over Z's, the retention times versus how we observed it. And as you might expect, what we do is we look at this and we take the couple or 3 peptides that give us the best response. Some of them are lower responding, some are okay responding, and then we have a few that we would call the best flyers. And along the way, we've acquired collision-induced dissociation spectra that tell us absolutely that we're looking at the correct peptide. We keep an eye on the retention times, and then sometimes we can gather some kind of argumentative information about how abundant or how detectable it is in different tissues. And so, but this idea that we put together a list of potential peptides to use, and then we test them experimentally and pick the ones we like the best. You can imagine we go through and we might have a hypothesis that increased fatty acid processing leads to oxidative stress. We look at mice that are had high fat diets. So it's just a matter of looking up in Wikipedia what all would be involved in the fatty acid processing. This gives us a list of proteins that we want to go look at, and we just one by one design methods for each of these proteins. And so the method for any one protein we call an assay, and then we build a panel of assays to cover this or some other biochemical pathway. And so we repeat that process as needed until we've built these things up. And this is a beta oxidation enzyme pathway. A couple of things you'll note about it is that we're a little bit liberal in sort of kind of how we define what might be a part of this. So, a couple of other proteins here. We started to get interested in peroxisomes. We included catalase in there, and then a couple of other proteases in there. We include bovine serum albumin in that. That's an internal standard for us, and so it's detected along the way. But this would be an assay panel. And this is what the data look like. So this is a selected ion monitoring experiment, in a controlled diet versus a lard diet versus a fish oil diet. So this is just one of my ACOX-1 peptides, that SFL peptide. ACADVL is the enzyme that catalyzes that same first step in the beta oxidation pathway. So this is a peroxisomal beta oxidation enzyme. This is a mitochondrial beta oxidation enzyme. And you can see predicted with finding the peaks. Skyline tells me which peak it is, and then determines the area under the curve. And again, a little bit like a Western blot, at least, if you kind of have an eye for chromatograms, you can see, lower abundance versus higher abundance, going from a couple to a couple and a half million in terms of peak height, up to 30 million in peak height. And, this is the way the analysis looks. With the mitochondrial version, again, 100 million, 120 million, 100 million, we can see that here in terms of different groups. This was an N=6 experiment, and because we know how much BSA we put in, we know how much protein we take for the analysis, we actually calculate a concentration in there. Some people might argue about the kind of the details of that, but I can argue back, I guess is what I would say. And so this is an experiment with 18 samples, control, a couple of different treatment groups, and I think this is at 20 weeks on the diet. And so, what do these data look like? These are spreadsheets, and so what you see here are for 3 peptides that we monitor for our internal standard BSA, the peak areas for all of those. We take the geomean of that as the response for the BSA. And then here are the 2 peptides for ACOX-1, the 2 peptides from ACA-DBL. Again, we take the geomean as what I call the total protein response. We normalize it to our BSA, and since we know the rate that the BSA was added, the concentration of the BSA, we can calculate the concentration of each of the analyte proteins. This is just a simple t-test at this point, that you see, essentially a doubling of the ACOX-1, 2.02, precise measures, 15% relative standard deviation, good strong t-testing, an experiment with N=6 in a mouse model. And even in the case of about a 34% increase, so this is 100 and whatever 23 times 100 is, increases, good precision leads to good statistical power. I point out here that ACOX-1 in heart is relatively low abundance, and just as a comparison, our friend GAPDH, which can also be measured in the exact same run, is about 500-fold more abundant. This was an 18 sample experiment. Each run takes about an hour and a half, so it's about 25 hours of LCMS time. And so, and you can imagine this is just a part of the spreadsheet. It goes on and on down through all of those proteins that you saw on the list. And so very kind of a speedy version of that, of kind of what we do with the targeted methods. And so, just a quick summary is that, we kind of turn the experiment around. We now go into these complex data sets and try and find and measure specific peptides as markers of specific proteins. And so what we really leverage in the mass spectrometry is the specificity of that detection, whether it be by very precise and accurate molecular weight measurements at high mass resolution, or by using the fragmentation pattern, and the fact that that fragmentation pattern is dependent on the amino acid sequence of the peptide. So specificity is the key to what we're getting out of that. Each assay is built based on known peptides that we can find that are linked to that protein being measured. As we do this, we inherently verify or validate that we're looking at the correct peptides. And then, the good news is we don't have to do this every time. That once built, an assay is good forever, can actually be transferred between labs. We build up this database that Skyline uses to show us where the proper peaks should be eluting. And then just a final point, I didn't really point this out very much, but that high resolution, that very simple 300 to 1100 scanning experiment, those data can be reinterrogated for essentially forever and ever. As new questions come up about the data, as you develop new assays, you can go back and it becomes a digital kind of archive of what the mass spectrometer saw in that sample, and that can be reinterrogated over and over again. We've actually had a couple of real life examples where we've been able to answer paper reviewer questions by going back and reinterrogating the data. We need to come back and revalidate, but we can reinterrogate those data. And so at this point, a speedy blast through that, but I'm happy to answer any questions.